This lecture is a review of the senses found in vertebrates. Now, nervous systems, and let's just take the human brain, for example, need information, right? So your brain has never seen your best friend. Your brain has never tasted lasagna. Your brain has never felt, your knee, your brain doesn't know where your body is. It must be told. And so senses then collect this information, some about the outside world, some about the inside world. Is your stomach full? Are you feeling pain anywhere? And then they bring this to the central nervous system. Now, there are both what are called general senses, and general senses are called general senses because, one, they can be perceived all over the body. So there's not one part of your body you feel touch from. You feel touch from here, pain, temperature, etc. So they're generalized. And they all project to the same region of the cerebrum, this somatosensory cortex. So here in the cerebrum, you have the central sulcus, and posterior to that, you have a fold, the post-central gyrus. And here you have a map of your body. You have a face area, a hand area, and thus you can know when something's touching my face, when something's touching my hand, because the general senses project to this region. Now, from an anatomy point of view, the general senses perhaps aren't the best places to start in a comparative vertebrate anatomy class, because many of them consist of single neurons. Some of these sensory neurons don't have any specializations in order to perceive a sense. So they're what are called free nerve endings. So, we simply have, dendrites without being wrapped in a capsule or anything else. And so some of these sensory neurons are perceiving, pain or cold or heat, or pressure, all sorts of senses. And obviously these go back to primitive animals. So there is obviously an advantage to be able to feel pain unless you could avoid a painful stimulus or any of these senses. And so we have free nerve endings. And then some of these sensory neurons actually do wrap inside, some sort of a connective tissue capsule. So there are collagen fibers, et cetera. And then these can contribute to, touch, to pressure, et cetera. And so these general senses then are difficult to study because we're looking at microscopic structures, and certainly invertebrates possess a variety of sensory neurons. The cerebrum was greatly expanded in the mammals, and so this somatosensory cortex, possessing a map of the body, is something that is primarily a mammalian phenomenon. In the most primitive mammals, the sensory and motor maps are together, and there is no central sulcus. But most of this lecture, I would like to focus on the special senses. They're special for two reasons. One, they are only perceived in one place of the body. We perceive light at our eyes, and not here at our elbow and here at our knee, etc. And then also they project to specific parts of the brain rather than the somatosensory cortex. The first one is the sense of smell. And so if you consider the human skull, we have this ethmoid bone with these tiny little holes in this flat cribiform plate. They are called the olfactory foramina. And it is here that the first cranial nerves leave what are called the olfactory bulbs and then go to an area of epithelia here in the nasal cavity where they will then bind to odorants. And so we have this olfactory epithelium where these bipolar neurons, so-called because they have two processes coming from either end, they then have a sensory cilium, which then has processes which can detect odorants. Now, it's interesting that these basal cells can produce new olfactory neurons, maybe at a rate of, say, once a month, because in most of the central nervous system, we never regenerate neurons. And here in the nose, we're regenerating them constantly. So that's interesting. Mucus is secreted here in this olfactory epithelium. This mucus both dissolves odorants so that these neurons can perceive them, but then will also wash the odorants away so we don't perceive them forever. And that's nice too. If you smell smoke or something unpleasant, you can avoid the room, but you don't have to go the rest of your life, smelling it, because it is attached there. And so these olfactory neurons then detect these odorants, and then they project to what is known as the olfactory bulb. It is a part of the cerebrum. It's a part of the cerebrum, but it is not the neocortex that's greatly expanded in mammals. It's rather the paleocortex. In more primitive animal, going back to jawless fish, the olfactory bulbs composed, about half of the cerebrum. So the cerebrum was to a large extent an olfactory place in ancestral vertebrates, but it has become smaller and smaller in humans. From this olfactory bulb, there is an olfactory tract, which then projects to regions of the brain. So once again, this is kind of a primitive part of the brain. It's what's called paleocortex, with fewer layers of cells than most of the cerebrum, called the neocortex. And it's fascinating for so many reasons. Smell is actually fascinating because once again, it is just different from the other senses. All of the other senses go to the brainstem and thalamus first. All right, and so the brainstem and thalamus, they filter stuff, whereas smell is coming directly into the cerebrum, and it hooks into areas which are involved in emotion and memory, like the amygdala, and so therefore, just smells often trigger memories. Smells often give us an emotional response in a way that sounds or sights don't have to. There's lots of sounds and sights that really don't get much of an emotional rise out of us one way or the other, but it's hard to say that with smells. And so once again, olfaction is fascinating. Now, in olfaction, it should be said, we vertebrates, we actually typically have two senses of smell. And perhaps, these buffalo sniffing each other, and the male buffalo will actually even sample urine, is a good demonstration of the second one. Mammals communicate because they often communicate with each other. Many mammals are social, and most mammals are actually nocturnal. All right, we think of the ones that come out during the day, but when you think of all of the rodents and the bats, which are actually, the big groups of mammals, the ancestral mammals were probably nocturnal, and the majority of mammals alive today are nocturnal. And smell was very important. Smell was not then important to communicate with each other. So when mammals typically sniff each other, whether it be a dog or this buffalo, they're getting information. They're getting information, say, about a female, whether she is an estrus or not. Male odors indicate his ranking in the group. Is this the alpha male that wins all his fights, the zeta male which loses all his fights? There are also other bits of information. Is this individual sick or not? What species does this individual belong to? And even how closely related is this individual to you? Because these odorants which we produce, these pheromones, are affected by our cell ID badges, the major histocompatibility complex, the MHC proteins. And in other words, there is another type of smell where individuals then produce body odor on purpose to communicate things about themselves to other members of this group. And in general, this sense of smell, which is perceived in a vomeronasal organ and is known as the vomeronasal system, this is important. But not so much in these gorillas, and not so much in humans, and not so much in any of the catarrhine primates. That would include the apes, like ourselves and these gorillas, and the old world monkeys, like rhesus monkeys, and baboons and mandrills and the like. And so why? Well, because there was a mutation in ancestral catarrhine primates, which inactivated this vomeronasal system. That grimace that that male bison was doing. So, one might think, oh, he just sampled urine, that's why he's grimacing. No, actually, he's grimacing so that that facial expression will better open the channel, which leads to this organ. So it's called the vomeronasal organ because it's in the vomer. Humans have it too. We still have a vomeronasal organ. It just doesn't work. The nasal cavity actually has two senses of smell. The primary function of this one is to perceive pheromones. In the catarrhine primates, it's mutated and it does not work. And so we don't get as much information from body odor as most mammals do, because one of our two senses of smell doesn't work. But it's very important for vertebrates in general. So for example, when a snake flicks out its tongue, it's trying to grab odorants and to then rub them against its vomeronasal organ. And so when we talk about smell, we humans, we have one sense of smell, this main olfactory system, but there are actually two in the land vertebrates. Now, when we go back to fish and study their genes, they actually had all different types of olfactory genes, including the ones which would help the tetrapods perceive odorants in air. That's a different group than the ones perceiving odorants in water, but fish actually had them, and they had vomeronasal receptors as well, but they weren't separated into separate systems here. Now, just one other comment. We humans and higher primates in general are not trying to use our senses of smell very much. The olfactory bulbs, they're small. The accessory olfactory bulbs, they're not even there anymore. They form as embryos, but then as the cerebrum expands, then they are lost. If you look at our genes, we have an incredible number of olfactory genes, over 1,000 for olfactory receptors. That's a really big number. Far more than the number of genes for light perception. But half of them are broken. Half of them are pseudogenes, mutated, non-functional genes. And so as we go up the primate family tree, we see more and more pseudogenes of these olfactory receptors as the primates began to rely more and more on vision and less on smell. Now, smell is so incredibly important, but obviously I'd like to move into others. But even for taste, 80% of what you think you're tasting is actually what you're smelling. An easy way to do this is, you or with a lab partner, go to your spice cabinet and say, oh, here's vanilla extract, for example, and say, oh, like my lab partner knows what vanilla tastes like. Do they? So ask your lab partner to hold their nose and then just, maybe get a Q-tip and stick it into, one of, these flavor extracts and put it on their tongue and say, what taste is it? If they're holding their nose, they would sense that it was wet, but it isn't until they let go of their nose that they say, oh, that tastes like vanilla. And then you realize, oh, wait, it doesn't taste like vanilla because I couldn't taste it with my tongue. It smells like vanilla. So actually, and not only do we smell things here, but actually going through the back of the pharynx, odorants in the air will then reach the nasal cavity that way. So then in addition to the sense of smell in the vertebrates, which once again is fascinating, but in our lineage has deteriorated, we have taste. Now, lots of organisms taste. It's a sense of chemo reception. But the taste bud that you see here, this was a vertebrate invention. No taste buds are known in other organisms, where you have this collection of these epithelial cells whose microvilli then have taste receptors. And when they are taste is perceived, then these epithelial cells release neurotransmitters onto sensory neurons. So taste buds begin in vertebrates, but they're not isolated to the mouth, because fish, I mean, they want to know what, the environment tastes like. So this is just going, lots of things taste, like, fruit flies and house flies, they're tasting their food, because each, fruit flies and, house flies, they're going after different types of foods. So fish, they are sensing this all over their bodies. And so if we were to look at, say, this lamprey, does it have taste buds? Yes, it does. And it has it in its pharynx, but it also has it over the rest of its body as well, its head and throughout the rest of its body. I mean, catfish are pretty impressive. I think catfish can have 100,000 taste buds distributed all over their body. So, catfish are tasting the environment, but they don't have to do it with their mouths. And these, starting with the jawless fish, the senses of taste, which we associate with salty taste, sour taste, and bitter taste, were known in vertebrates. When we get to the bony vertebrates, the osteichthyans, they can sense sweet taste as well. By the time the sarcopterygian fish evolved, these would be the ancestors of the tetrapods, taste buds were now limited to the mouth, tongue, and pharynx. And in humans, they're found on all of these places. When we're young, we're very sensitive to taste. We have, taste buds all throughout our throat, but they tend to decrease as we age and are then primarily on the tongue. Now, speaking of tongues, do fish have tongues? Technically, yes, but it's a different type of structure than what we associate with tongues. They're made by different embryological structures, and they don't have muscles, or many muscles, so they're not very mobile. So when we speak of tongues, this was kind of the invention of the tetrapods, which live on land. So tetrapods develop a true tongue, and mammals take this to the greatest developmental stage, where 5 different embryonic structures fuse. A lot of the muscles supporting the tongue are coming from the hyoid bone. One of the reasons for this is because mammals need so many calories to get their warm-blooded, big-brained bodies through a day. They're going to chew their food so that they can, get every single calorie and nutrient out of this food. And so therefore, because food is spending so much time in the mouth, then taste has more of, a chance to be effective. If you think of like an alligator or a lizard, if you're simply going to chomp, chomp, and swallow on your meal, then the sense of taste is less significant. Now, I had mentioned that for the most part, a lot of what we think we are tasting is actually our sense of smell. Taste just has some basic questions it needs answered before you decide to swallow something. Because if you make a mistake, that could be a problem. I mean, because you could get sick, it could be poisonous. So, while, our sense of smell is wondering what kind of cheese is this, our sense of taste is much more basic. So it's, asking just basic questions. Is this substance sweet? Because that's good. That's calories that I need to get through this day. Is this substance salty? We like salty foods because, especially if we live far from the ocean, salts, aren't necessarily easy to come by, but we need ions for our nervous and muscular systems to run. And so, therefore, if we lose ions through sweat or urination, we need to replace it. So here is just a quick demonstration. I apologize. So not only can we smell food coming in our nose, but also odorants in the oral cavity from what we're chewing can then enter the pharynx this way and be smelled. That's what's called retronasal olfaction. And so we and so salty foods we like. We sense the presence of certain amino acids, and the name of this taste is umami, which is good because there are essential amino acids which our bodies cannot make. And so therefore, for protein in our body, if we're going to, so not only sweet taste, which would give us, the ability to continue to generate heat, but also, if we're going to, build muscle and build protein, we need to know where the amino acids are because our liver can make some of them, but about half of them we can't make. So those are good tastes, and we tend to like those tastes, and we want more of them: the sweet, the salty, and the beefy, a taste of amino acids. But then there are tastes which we don't like. We don't like sour tastes, which is good because sour things have quite likely spoiled. They've been around for a while, and things which are sour, then they might have bacteria, they might have nematode worms, which are parasitic, and so by avoiding sour foods, we're probably avoiding getting sick more. And then we also sense bitter tastes, which is also good because a lot of the bitter compounds we taste are what plants put in their food to kill animals, or at least deter them from eating the plants. Plants don't like it when animals eat them. And so plants commonly put really nasty things in their leaves to deter animals from eating them, or to punish them if they do. So they're poisonous. And so things that taste bitter to us have plant poisons in them. Now, some things taste mildly bitter, like broccoli or coffee. Are there poisons there? Yes. Our liver takes care of them, though. And so, there have been foods which have been cultivated for humans. But once again, here are these amino acids, half of which we can't make. They are what are called the essential amino acids. So we like the umami taste. So taste is more basic than smell. Taste is simply asking the question, now that you put this thing in your mouth, is it a good idea to ingest this? Is this good food? Does it have calories? Does it have amino acids? Does it have ions that you need? Or is it something that is bad and you don't want to take into your body? So is it sour and might get you sick? Is it bitter and might be poisonous to you? There are 3 cranial nerves which process taste. The region of the cerebrum which processes taste is at the bottom of the somatosensory cortex. And then there are, so it would be there in black, there at the bottom of the somatosensory cortex, and the we have the senses of the head reaching the thalamus, where they then get projected to this area of the somatosensory cortex here to give us that sense of taste. The third of the special senses, once again special because they are perceived in one place of the body and go to a special part of the brain, are the senses of the ear. And here I have to lump a whole lot together. So just to begin with the human anatomy, we have an external ear made of the pinna and this external, this auditory tube going through this external auditory meatus. We have a middle ear, which connects to the throat. And then we have an inner ear where the senses are made. So this connection to the throat, can change in embryos as skull bones fuse and little pieces come together and become one larger bone. In some fish, the swim bladder can actually communicate with this area. And so in humans, this tube is good because it helps to balance air pressure on either side of the tympanum. So it's called the pharyngotympanic tube. But once again, it's kind of dependent on how skull bones come together, which obviously vary in the and over the course of vertebrates. Now, the sense of smell, the senses of the ear rely on these cells called hair cells. So whether we're talking about sound detected in the cochlea, or balance in the semicircular canals, or linear acceleration or the pull of gravity in the the regions of the vestibule known as the saccule and utricle, they all rely on these hair cells, where we have a big cilium known as a kinocilium, and then these other structures called stereocilia, which aren't actually cilia, they're actually microvilli, which are stiffened with actin filaments. This is what's called a mechanoreceptor. Its job is to sense this bending. There's a distortion. All right, so these, cilia, the kinocilium and the stereocilia, they bend. This changes the amount of ions which come into this cell, and then the electrical change in this cell then causes neurotransmitters to be released onto sensory neurons. So now the brain can be informed that there has been a change. And so these hair cells, they are the basis of the senses of the ear, which I'll get back to. Now, all of the components of this hair cell predate the vertebrates. So invertebrates can have cilia, obviously. These go back to eukaryotic cells. Cilia can be sensory. Cilia can be associated with microvilli in order to function as mechanoreceptors, et cetera. So all of the components are not unique to vertebrates. Now, it is going to be the chordates which have hair cells. But interestingly, even though the sense of hearing in humans and the sensing of hearing in flies are not directly correlated, nevertheless, they are sharing some of these cellular structures, so that mutations in the same genes can actually cause deafness in both humans and fruit flies. And so that's interesting, indicating that some of these aspects of kinocilium or the perception go back before chordates. The only hair cells are known in chordates, starting with the invertebrate chordates. So like these tunicates, they possess true hair cells, which would be the senses of the ear, but they're not functioning in hearing in these tunicates. They're functioning as mechanoreceptors, although they also then do have a gravity sensing structure in their head region, which has genetic homologies to the ear of vertebrates. All right, and so hair cells and a potential otic region exists in the chordates. By the time we get to the vertebrates, we then have hair cells functioning in groups. So hair cells, they can function by themselves and just be a mechanoreceptor. But what we're more interested now is bunches of them together called neuromast organs. So you have a bunch of hair cells all together. Very often this can be encased in a dome of some sort, a cup known as a cupula. And so therefore this just increases its sensitivity. Anything which causes movement here, which distends the gel of, say, this cupula, will then bend the stereocilia and kinocilia and then be perceived. And so this is a neuromast organ. Now, what would distend this area? It could be something from the outside world, like water, like if a fish is swimming and there's a predator or prey nearby, or just water pressure from current, etc. This can push this structure and be perceived. But in my ear, these neuromast organs are an enclosed cavity, so they're not exposed to the outside world. But now there's fluid in my ear in certain channels, which can then be distended. And so various things can move this. So I know, we want to talk about what we find in the human ear, the cochlea where sound is perceived, the semicircular canals where balance is perceived, or the regions of the vestibule where we sense acceleration. But we should actually begin with another structure known as lateral line organs. If you look on the bodies of most fish, on some it's more obvious than others, you can see pores, and very often these pores occur in a line. If you look closely, there's a pore, a pore, a pore, a pore, a pore. So there's a line here. There's another set on the snout there and there. These pores let water into a groove where neuromast organs are. So there could be like hair cells right underneath the skin there and there and there and there. And so there's this groove along the body, which has lots and lots of hair cells. Collectively, it's called a lateral line system. When you look on the outside of a fish, sometimes their coloration is just a little bit different along this lateral line structure. So here, for example, on this catfish, you can see these pores forming these lateral lines, some on the head, some is studying the rest of the body. On this carp, you can as well. So these hair cells are functioning in fish, but they are functioning to detect water. So once again, as water moves, it's going to displace those cilia in the neuromast organs interior here. And so thus here you can see pores, but also you can kind of see like a slight depression on this shark where the lateral line is. Once again, sometimes you can notice, there'll be changes in pigmentation along here, etc. But so fish use these. Some fish will have them, there are scales which have pores in them. So even though a bony fish is covered with scales, it could still have water coming into these lateral lines. And thus, as a result, when the water moves, or when fish are moving nearby, fish that might be prey or a predator, that's just a sense that fish have using this lateral line system. Now, fish use lateral lines. If you look at the skulls of the early amphibians, they have little grooves where these structures were. And so the first amphibians as adults were using the lateral line system, which makes sense. They were descended from fish. And you can still find it in aquatic amphibians today. So some larval amphibians, some amphibians which are aquatic as adults will retain lateral lines, but the tetrapods, which are terrestrial, so that would be terrestrial amphibian adults, but that would then also be then reptiles, birds, and mammals no longer have lateral lines. But that's important because from the lateral line system comes the senses of the ear. So the vestibule in the ear, which is where balance and gravity are sensed, is derived from a lateral line system. So you have all of these neuromasts function in the lateral line system. But then one of this area got better at being more specific towards gravity and balance. Now, I know this sounds odd because in our ear, this area is for hearing and this area is for balance. But lateral line systems are more generalized. It's thought that lateral line systems can actually perceive sound a little bit. So, fish, because what would sound be underwater? Sound would be, there would be, vibrations in among water molecules, which then would be, sensed by this lateral line system. Sound was thought to be originally, perceived by lateral line system, but these lateral lines in the head, they formed this vestibule region. Now, in humans, the vestibule has the semicircular canals for balance, but also these areas for acceleration, whether it be vertical or linear. And so it was a derivation of the lateral line system, which gives us the vestibule, and then later a modification of a region of the vestibule, which gives us the cochlea, where sound is perceived. Now, humans have 3 semicircular canals. These help us detect balance. So there's fluid here, all right? And so when I move my head in this plane, or this plane or this plane, it causes movement of fluid in these semicircular canals, which then can be perceived by the hair cells here in this cupula. And so hagfish have one single semicircular canal. Lampreys have two semicircular canals, and then the jawed vertebrates have three semicircular canals. So that would first have appeared in ancestral jawed vertebrates, like the ancestors of modern sharks, and then this is also then present in the tetrapods and amniotes and mammals, etc. So the semicircular canals, hagfish have one, lampreys have two, the jawed vertebrates have three. The pores which let water come in, they still open into the vestibule in sharks. So in the jawless fish and sharks, it's still water from the outside world, which can have an influence on the senses of the vestibule. Meanwhile, in the bony vertebrates, the bony fish like this one and tetrapods, the neuromasts of the inner ear are completely enclosed. The outside world is not letting in water, or a direct stimulus. Here, there are, closed channels in the ear here. Now, this otic region starts to become more and more important in jawless fish. There's just these little pieces of cartilage here and there of the chondrocranium. But obviously, when we look at sharks, this is becoming more prominent. And so, as we consider sharks, so here in the lamprey, they don't have much cartilage. But here is the chondrocranium of a shark. And you can notice the very prominent otic region, the otic capsule, the cartilage which encloses the ear region, which would include three semicircular canals, which is typical of all jawed vertebrates. The jawless vertebrates have fewer than three. So here, once again, the cartilage of the otic capsule that encloses the ear region here. Obviously, we are interested in how humans hear and maybe the progression of the evolutionary changes in the human lineage. But the senses of the ear and related lateral line system, they vary enormously with that throughout the fishes. So when you look at the number of cranial nerves going to the lateral line system, so we land very, there's the 12 cranial nerves and we number them. Well, I mean, if you include fish, I mean, there's all of the cranial nerves going to the lateral line system, which varies a great deal. One of the things which is interesting in one big group of fish, which includes the catfish, is that the swim bladder then communicates with the ear region and helps in the detection of sound. And there's a bunch of little bones known as the Weberian ossicles, which help this, but they vary enormously, not only among fish, but also like among catfish, the degree to which this happens. And so, this is a very generalized lecture. And if we were to, consider, the variations in fish, that would be much different. So here's just, this is unrelated to hearing, but look at these catfish brains. Look how different one catfish brain could be to another. So when we start talking about this Weberian apparatus for hearing, that varies enormously, from fish to fish. But fish make it on to land, and thus then there's a challenge because hearing underwater is very different from hearing on land. But the Sarcopterygian fish, which were ancestors of the land vertebrates, they were adapting their ears to hear on land. Now, if you were to consider what happened over, a couple tens of millions of years, as this Sarcopterygian fish, type of thing, evolved into, say, this tetrapod. So that was, a fish like Euthanopteron becoming a fish like Ichthyostega. Oh, there were so many, changes going on with the skull, so many involving senses, like the eyes were moving so you could better see on land here. But if you'll notice that a lot of them have a notch in this region, that's the region of the ear. And so the fish, they were changing. They were losing their opercular bones that support the gills. And they were now modifying the area where they could hear. So here's a sarcopterygian fish. Something's going on there, a change from the normal fish skull right in the region of the ear. If we look at the middle ear space, you can say if this was, say, an earlier sarcopterygian fish, Euthanopteron, and this is an amphibian, Acanthostega, one of the most primitive ones. Here you have a fish known as Panderichthys in between the two, and you can see that a lot of the changes to the ear of the first amphibians were modifications which occurred in the early sarcopterygian fish. And so there are, I'm sorry, that was the wrong button. And so hearing on land is different than hearing in water, but the changes were started actually in the sarcopterygian fish. Now, it would certainly be easier if we could say, the first amphibians, finished that, and when an amphibian hears this noise, it's doing it in the same way as we do. The reality is it's not that easy. So for example, let's take eardrums, all right, the tympanum. Frogs have them, obviously. They have that really big tympanum. All right, we'll see in a second. But salamanders don't, and snakes don't. And so it turns out that the ear was not done in the first land vertebrates. And so therefore, as we look at the descendants of diverse lineages of tetrapods and amniotes, we'll see differences because the ear, matured differently. So here you can see the big tympanum here. The amniotes did make some changes. So there's a tube, the inner ear is now deeper in, so there's a tube connecting it to the outside world. And so this external auditory meatus is an opening, and that tube lengthens. The region which detects sound. So in fish, the lateral line system probably helps with sound, but the vestibule certainly does. And the vestibule doesn't do sound, it's doing balance and acceleration, but in fish it can do sound. But there was a region of the vestibule that started to specialize in only being sound. It's called the lagina. And in mammals, it coils and becomes what's called the cochlea. And so in amniotes, this structure gets longer, and in mammals, it will become more coiled. And so there are certainly changes which are shared, but the reality is, well, if we were to talk about a tympanic ear, where there's a tympanum, an eardrum, which vibrates, and then the inner ear is completely enclosed, that type of ear actually evolves separately in different lineages, and multiple lineages of reptiles, in the lineage of amphibians, leading to the ones alive today, and then in mammals. So once again, the structure which mammals have called the cochlea in amniotes is also long, but it's uncoiled. So here I've just uncoiled it, just to talk about it. But it would be the mammals which would coil this, and I'll talk about how it functions in just a little bit. Now, one of the important things in the ear is that we have these three little bones in humans, the malleus, the incus, and the stapes, which take the vibrations of the eardrum and they amplify them about 20 times so that by the time they are transmitting these vibrations to the inner ear, they're bigger, which greatly enhances our sense of smell, of hearing, of sound. So there are these auditory ossicles, but only mammals have these three. The first one, the stapes, is the oldest. So here's the three little ear bones which occur inside the middle ear of mammals. So the stapes here in pink used to be like just a regular skull bone, and it was big and stout, and it functioned in support. So it functioned in support, but also began to transmit sound. So vibrations of the perceived in skull bones could then be perceived as sounds. Now some of the reptile of the amphibians. So here's an amphibian which seems to be a little more closely related to the reptiles. Notice here that the pink bone has become much smaller. So it's this stapes, then as time went by, and some lineages served less and less of a role in support of the skull, and more of a role in hearing. And so there is, this one, ear bone, then auditory ossicle, the stapes. But as one then goes from the primitive pelycosaurs, these would be synapsid reptiles, which would be the ancestors of mammals, we look at the lower jaw, which is fascinating. And so I have a video just on that. But the original jaw joint, so when reptiles open and close their mouths, when the ancestors of mammals opened and closed their mouths, it was where this red bone touched the yellow bones, and this pink bone is also of interest as well. Over tens of millions of years, as the ancestors of mammals went through various stages, pelycosaurs to therapsids to cynodons. This jaw joint between the red and the yellow bones, what's known as the quadrate and articular, this then changes because eventually mammals don't open and close their jaws with these yellow and red bones. In mammals, the blue bone, the dentary, touches a part of the temporal bone called the squamosal. So mammals have a dentary squamosal jaw joint when we open and close our mouths. And so over tens of millions of years, this jaw joint gradually got smaller and smaller. These bones became less prominent as mammals transitioned into this new one. For a while, there were two jaw joints, both here and there. And so mammals were using the old reptilian one and the new mammalian one. But at some point, then the reptilian bones were no longer needed in the jaw joint. And so lots of bones were lost in the evolution of the lower jaw of mammals. But then the question then comes, what do you do with the little yellow-red bones and the pink bone as well? Well, you could lose them, but mammals did something interesting. Mammals then, as little separate bones came together and fused the temporal bone. So the temporal bone came together from pieces. So there was a squamous portion, a petrous portion, which were separate bones, the squamosal and the petrosal. They smushed together and formed the temporal bone. But where they smush together, they leave a space. And that space is where the inner ear is. But then just prior to the inner ear, there's a middle ear, which not only has the stapes, that was an auditory ossicle inherited from the early tetrapods, but now those yellow and red bones end up here. So here you can see the jaw joint, the big blue dentary bone, which becomes the only bone of the mammalian lower jaw. But these little yellow and red bones, they were once associated with the jaw region. They just migrate into the middle ear, where they then serve as the auditory ossicles, which only mammals have. That pink bone kind of forms a hook. It's called the angular. It then supports the eardrum. So only mammals have the incus and malleus. These are derived from those, the quadrate and articular bones of the reptilian jaw. And so if you were to look at a marsupial embryo developing, these bones begin in the jaw. and then migrate to the ear after they've been born and are maturing in the pouch. If you were to look at a human fetus, the angular bone supports the eardrum. It's the separate bone, which then just kind of becomes one with the temporal bone. So you can see the angular bone. And then in the ear, then are the remains of the quadrate and the articular. Once again, if this is the jaw, these bones were forming in the jaw region, but now they have been modified to be part of the ear. I'll talk about the ear functions in just a second. Obviously, then different lineages of mammals have done different things. One of the most obvious ones is changes in the elastic cartilage known as the pinna or auricle here, which then directs sound to the auditory canal. And so obviously the amount of cartilage in the pinna can vary, as you can see here in, this elephant, composed to, like say, a cat or a human. The most significant differences would be in bats. Bats have crazy variations in their ears for a reason. While the first bats and the Microcureopterans today. So there are bats today which can come out during the day and they can hunt for fish or they can hunt for fruit, etc. From these generalized bats came another group which then composes the majority of bats today, the Microcureopterans. What they do is they emit very high-pitched frequency sounds out of their noses, and then as they bounce off objects, they perceive them with their ears, and thus they get a sense of direction. They know not to fly into a tree, but they can also detect where flying insects are. And so once again, ears, have obviously been evolving for lots of great periods of time. And it wasn't done with the first mammals. And in some lineages of mammals, like, bats, a fascinating discussion could follow about then how bats are using their ears to more or less see at night with. So they can, find bugs, they can avoid flying into things because of the precision of how they're perceiving sound. Just to, kind of circle back to the human ear. And so our cochlea is a coiled structure which has then all of these hair cells in it. Vibrations in air molecules cause the eardrum, the tympanum, to vibrate. Those vibrations are then amplified as they pass over these auditory ossicles, the malleus, the incus, and the stapes. Once again, only mammals have the malleus and incus, so that the vibrations pushing here are 20 times bigger than they otherwise would have been. So this will push against our oval window that causes a bulge coming out of a round window, and this causes fluid to move in the cochlea. So if you were to take that wound structure and unwind it just for simplicity's sake, you could see that the hair cells are in the middle along a membrane, right? So if we were to unwind it here, as we push, we'll cause a current in fluid moving this way over the membrane, which holds the these hair cells. And as sounds cause vibrations, and that displaces this, that causes those kinocilia and stereocilia to move, and thus this can be perceived as sound. A loud sound would move lots of rows of hair cells in a certain region, while a soft sound would just move a smaller number. And so your brain can distinguish between loud and soft sounds depending on where, depending on how many rows of hair cells are displaced here. And because this area is thicker, so this area of the basilar membrane is thicker, only a high-pitched sound has enough energy to displace this area. low pitch sounds, they have less energy, so they would only be able to displace the apex. And so your cochlea then can not only distinguish between sounds of different volumes, because that will change the number of hair cells moved. So here is it, still coiled, here is it uncoiled. And so as fluid goes through the ear, so when sounds push the tympanum, which pushes on the auditory ossicles and causes then fluid to move, it'll displace this area and then cause these hairs on the hair cells to move, which they then communicate to the sensory neurons. So the number of hair cells moving will correspond to loud versus soft sound. And then a high pitch sound, if you're singing a high note, that would displace this area most, or if you're singing a low pitch sound, that would displace this area most. And so your brain would know the pitch of the sound based on where the sound was perceived. So I go into that in a little bit more here. So here the cochlea detects sound. But remember, the ear is more complex than that. We also have these 3 semicircular canals. And when we move our body, certainly our head, because the ear is in the head, but the head's connected to the body. So when you're, moving this way or that, your semicircular canals get a sense of balance based on the movement of fluid in the semicircular canals and the displacement of those hair cells here. And so thus these participate in balance. Once again, in fish, sound is also perceived here. All right, but you get an idea of balance. And then finally, in this vestibule, there are 2 areas where the hair cells rest in a membrane on which there are these little crystals, these little calcium carbonate crystals known as otoliths. And what happens is when we accelerate, there's a bit of a drag on because of inertia. So I indicate this with a car. So if you are in a car and the car accelerates, we'll get there in a second. So the car goes forward, but your body goes behind. That's inertia, a resistance to movement. And then when your body goes forward, there is inertia that pulls on these otoliths. And so if you were to accelerate, whether in the linear sense or an elevator going up and down, that would then cause these little crystals to just lag behind the rest of the body. This bends the membrane, which bends the kinocilium and stereocilium, which then you can detect as acceleration, both linear acceleration and vertical. We have these 2 different regions in the vestibule which sense these. So the 8th cranial nerve has the name the vestibulocochlear nerve, because not only are you getting a part from the cochlea where sound is detected, but also another portion from the vestibule, which is sensing balance from the semicircular canals, and then also that linear and vertical acceleration from the vestibule. Vision is a very important special sense. And in one sense, just looking at the outside of the eye, as I've mentioned before, all of the jawed vertebrates, and to some degree in the jawless vertebrates, we see a constant state of eye muscles. So there are 6 eye muscles of the jawed vertebrates, which develop from 3 different embryonic blocks in the developing head. And the same 3 cranial nerves innervate them. So we have the ocular motor, the trochlear, and the abducens nerve innervating these 6 muscles in the same pattern. And so the eye is a very old structure, and all of the jawed vertebrates and to a lesser extent in lampreys, we can see a conservation of these eye muscles. So these are among the most conserved muscles in the vertebrate body. Let me just start with the human eye. We split it into layers. So there's an outer layer called the fibrous tunic, which is made of collagen, forming the white of the eye, the sclera. But obviously we have to let eye into the eye. So part of it then is transparent, forming a cornea. There are proteins which are transparent. They are meant not to interfere with the passage of light, called crystallins, which allow the light to come in, and the cornea is bent so that it can help bend light and focus it. So here you can see the next layer, a vascular tunic. That is where the blood vessels are, but also notice all of the melanin. So it's darkly pigmented here. And this is, part of the vertebrate eye, a pigmented epithelial region associated with the retina. This melanin helps to absorb light so that it's not bouncing around and interfering with the perception of vision. Anterior in this area, there is what's called a ciliary body, which has ligaments. The ligaments attached to the lens of the eye, and then they can pull on the lens, as we'll see in a second. Finally, there is an iris, which has muscle, which can then constrict or dilate and control the amount of light that comes into the eye. Because if too much light comes into the eye, it overwhelms the photoreceptors and you're blinded. If too little light comes into the eye, then you can't see. So you want just the right amount of light to come into the eye. So the smooth muscle of the iris then changes the diameter of the pupil and thus controls the amount of light coming into the eye. Here is the lens being supported by those ligaments from the ciliary body. Now in humans and in the higher vertebrates, the way that light is focused is that if you're looking at something that's far away, then the light from that faraway object is already coming in more or less in parallel to your eye. So you don't have to bend it as much to focus it on the retina. And so if you don't have to bend it that much, it doesn't have to pass through an awful lot of lens, stuff. And so therefore the lens is then pulled taut. There are muscles in the ciliary body, which then pull on these ligaments. The lens gets thinner because you don't have to bend the light all that much to focus on the cow. But if you're focusing on something which is, close to your face, you need to bend the light more, in which case the muscle here relaxes, the lens gets fatter, and then you can focus on something which is now close to your face. As I'll, go through in just a minute with other organisms, that evolved later. So lampreys, what they don't change the shape of their lens. What they do is they push on the eye and they change the, so if there's a corneal muscle which pulls on the cornea and then can change the shape of the eye, which then changes the position of the lens. In things like sharks, there's muscles here which can move the lens forward or back. So you're not changing the shape of the lens, you're changing the position of the lens in order to focus. So going back to early, well, I'll get back to early in a second. So in the human eye, we have both what are called rods and cones. The cones are so called because the primary cilium, which has the photoreceptor molecules of opsin, they are cone-shaped. And there are three different kinds in humans, one which responds best to red light, to green light, and to blue light. And so thus we have trichromatic vision, distinguishing between those three colors. Now, in an anatomy class, the molecular biology of, rhodopsin is a bit beyond us. But just real quick, this is a type of protein called a G protein coupled receptor. Interestingly, the receptors for smells in the nose and taste in the tongue are also G protein coupled receptors. But this one's called opsin. and it interacts with what's called a chromophore called retinol, derived from vitamin A. And essentially it's this: when light hits retinol, it will change its conformation, it changes its shape. One shape sticks to opsin, one shape doesn't. And so therefore, the number of opsin molecules which have retinol attached is then dependent on the amount of light which has reached that molecule. So when light reaches it, it causes it to dissociate or bleach, because this pigmented molecule is then leaving. And so then if the photoreceptor, the cell, changes the amount of neurotransmitter it makes based on how much of this opsin is bound or not, this then indicates that this photoreceptor is telling the brain when light has been perceived. So at this point, I'd like to just kind of consider where all of this comes from. Opsins, G protein coupled receptors, go back to bacteria. So these are in all living things, as are opsins. Opsins are ancient proteins. Now there are different groups of the opsin family tree. Some opsins are only known in animals. Some are only known in certain animals. And so talking about opsins or even crystallins, the proteins which make up the lens, they predate lenses. So yes, they're transparent, and that's great for use in the lens of the cornea. But the reality is that they are modified versions of enzymes or heat shock proteins that had other functions. And so some of the most important molecules in the eyes, they go way back. Opsins which bind retinol, these go long predate the vertebrates. Fruit fly eyes are obviously very different from human eyes, but fruit fly embryos use some of the same genes to essentially say, put an eye here, that human embryos do. So there are lots of different types of opsins found in animals, and so these go way back, as do some of the genetic transcription factors, which say put an eye here. So that brings us to who were the ancestors of the vertebrates. Well, it was those invertebrate chordates, of which we still have a couple alive today, not the same ones which give rise to the vertebrates, but hopefully we can learn a little from them. Do they have big, complex lateral eyes? They do not. But that's good, because they don't have big, complex brains. They wouldn't be able to process vision per se if they did. And so they have opsins. They have a number of the eye-specific genes. They have photoreceptors, but they don't have big lateral eyes. They do have both what are the cone opsin gene and the rhodopsin genes. So cone opsins, so we associate them with color, all right, because we have different kinds of color. But if you were to just have one, this allows you to perceive things in bright light, whereas the rod opsins are more sensitive and they bleach, and they allow you to see things in dim light. Obviously important for many mammals which are nocturnal, but obviously a lot of fish, they are living in the deep. So by the time of the early vertebrates, there were both photoreceptors which could respond to bright light and to dim light. Now, these photoreceptors, these rods and cones, it is their primary cilium which has the opsin proteins in it. So almost all of the human body cells have one cilium, a primary cilium. And in some, this is modified to be sensory. And so that's one of its functions. These primary cilium, they're very interesting. They express receptors, they are involved in some signaling pathways. But in the eye, this is this structure here. This is a modified primary cilium, just as this was in the olfactory olfactory neurons. And so This is where we detect light. And this type of receptor then goes back to these organisms, but it kind of reminds us more of the pineal gland, which I'll get to. Now, so if the ancestors of vertebrates didn't really have like big lateral eyes, where do eyes come from? Well, interestingly, from the brain. All right, so what is a transition to an eye? All right, well, there's lots of different answers because in the animal kingdom, there's different kinds of eyes. But the vertebrate eye, what's a good if you want like a halfway point, if you're asking how could this evolve, what is like a halfway point to an eye? And the answer is a photoreceptive part of the brain. Now, what I just said sounds odd, but let's just, speak anatomical truth here. The retina is part of the brain, all right? We don't think of it that way because it's left the brain. We call the axons which lead from it a nerve. It's not technically a nerve because a nerve connects the brain to something outside the brain. Because the retina is technically part of the brain, it is better called the optic tract. And here you can see a pig embryo developing. Notice that the optic cup where the retina will form, this is derived from the brain. Here's the brain. All right. So what happens is as the skull bones form, this moves outside the cranium so that the bones can protect the brain, but that the eyes can still perceive light. But the retina develops as part of the central nervous system, as you can see here. Here's the brain. Here's the optic cup forming. There's the lens, etc. So the optic nerve, technically, it's not a nerve. All right, because it's connecting this part of the brain to the retina part of the brain, which just got displaced during embryological development. So I say that once again because the transitional form of the eye is a photoreceptive part of the brain. So let's go back to those chordates. Do they have big complex eyes? They do not. But they do have these photoreceptors, okay? And by the time we, here's a tunicate, it has them. And they are expressed in the brain. So originally it was just the nervous system in general, not just the eye, but lots of places in the nervous system which detected light. Now, we would eventually get things in the eye called rods and things in the pineal gland, which I'll be talking about, called pinealocytes, which are related to rods. But these early chordates, they have lots of structures. There's different places in the, oh, I thought I was getting in this video. All right, so next one. There's different places that are detecting light all throughout, say, a lancelet's nervous system. You're looking at spinal cord. It's got lots of photoreceptors. So what we think is that the ancestor of vertebrates did not have vision. It didn't have eyes for vision, and it certainly didn't have a brain capable of processing vision. What it did have was photoreceptors in the brain, probably the beginnings of a retina. Maybe there were lateral structures or not. If not, then it was just an ancestor of the pineal body and these chordates, and the retina comes in the first vertebrates. And rather than function in vision, this was functioning in other tasks. Now, I know that sounds odd because, once again, a lot of people will ask, well, if you don't have an advanced eye, how can you see? What would be a transitional form? You're like, that's good. They might say like, oh, there's no function that you could have. Well, to be perfectly honest, the eye is a pretty complex place. And there's lots of different layers of cells here. Bipolar cells, horizontal cells, there's ganglion cells, which I guess we'll see in the next video. They express different opsins. They perceive light, but they don't do vision. So there are lots of reasons to perceive light, not just to see. You want to know if you're a fish, where the surface is, where the deep water is. You want to know how long the days are. You want to know what season it is. You want to know whether it's day or night. All of these are very important, and they aren't all vision. They're just perceiving light. And so there are different kinds of opsons. Some opsons don't do vision. They're perceiving light for these other functions in the brains of these primitive things, but even in our own eyes. We have cells perceiving light which are not contributing to vision. They are contributing to like biological rhythms based on the length of the day, etc. Okay. By the time we get to the jawless fish, they have eyes. So here's a lamprey with a really nice eye there, and it's clearly homologous to ours. Now, it's not exactly like ours, obviously. So if we were to say, look at the eye muscles, there are similarities, all right? But then there are also differences, and we could argue as to whether this muscle is homologous to that muscle, et cetera. The eyes of lampreys have rods for dim light and cones for bright light. And lots of other things are similar, like the shape of these photoreceptor cells, just lots of their molecular biology. The retina has 3 layers. They can even discriminate between different intensities of light and perhaps even different colors. There are differences here, like the ganglion cells, et cetera. They can change the diameter of the pupil to change the amount of light coming in. They have crystalline in their eyes. So lampreys have eyes which are directly comparable to those of the later vertebrates. This raises an interesting question. Here's the second type of jawless fish, hagfish. What are their eyes like, and are they interesting? And that's a tricky question, because the question then boils down to, where do hagfish fit in the vertebrate family tree? I'm sorry, before I get to that, let me just go through the hagfish eye. Hagfish are jawless fish like lampreys, but their eyes are very simple. They have no lens, no cornea, no iris, no pigmented epithelium. You remember that black region with all of the melanin. They don't have extrinsic eye muscles that you just saw in the lamprey. The retina has 2 layers instead of 3. They have this weird thing where the eye is actually covered by translucent skin. No other fish has that. The retina projects to a different part of the brain. The photoreceptors look more like the ones we find in the pineal gland rather than those of the rods. And the eyes, they don't do vision. They're doing those other tasks like, where is up and the biological rhythms. So why do these hagfish have such simple eyes? And so one possibility is, well, their ancestors had big eyes or bigger eyes, and that they have simply degenerated. And so a lot of molecular evidence tends to say that hagfish and lampreys are sisters. They are equally related to the jawed vertebrates. So that molecular evidence supports the idea that hagfish eyes are simple just because they've broken down or they've degenerated from, just like snakes have lost their limbs, and that some blind cave salamanders have lost their eyes. Here are, a group of fish which reduce the importance of eyes. And there is a fossil hagfish which seems to have more prominent eyes. And so, maybe, hagfish eyes have degenerated, in which case they're not all that interesting, at least not in an evolutionary sense. A second explanation is that hagfish and lampreys are not sister groups. So if you compare genes, the genetic analyses have tended to show that you would have had the first vertebrates. They were jawless, and they would be the ancestors of both lampreys and hagfish, but then one lineage then became the ancestors of the jawed vertebrates. In this one, lampreys and hagfish are equally related to the jawed vertebrates. However, instead of molecular evidence, the older way of classifying this based on anatomical evidence, oh, and in this in this system, if you ask where did the lens, cornea, et cetera, the complex eye evolve, it evolved here, and that hagfish just lost it. So the ancestors of hagfish would have had more complex eyes, but they just lost them. Earlier, the way of classifying hagfish was that hagfish were what were called craniates, that lamprey and everyone else would be vertebrates, but that hagfish were craniates but not vertebrates, that hagfish would be slightly more primitive than jawless. And there's a whole lot of anatomy to support that. The hagfish, they have the most primitive hearts. They have the most primitive brains. They lack a cerebellum. They have the most primitive kidneys. They're still using their pronephros. The semicircular canals, they have one, while lampreys have two. In system after system, hagfish anatomically seem simpler than lampreys. If they then had a position in the family tree here, what that would mean is now all of a sudden those simple hagfish eyes become rather interesting, because maybe this is the eye structure of the first fish, and that the lens, cornea, iris, etc., the larger, more complex eyes evolved here to be the common ancestor of the lampreys and the jawed vertebrates. So hagfish eyes are extremely simple. How interesting are they? Well, that depends on whether you're classifying hagfish as a sister group, meaning that their eyes are just, a degenerated version of what their ancestors had, in which case not all that interesting. But if this represents their position in the family tree, their eyes might represent, more what the first vertebrates had, which makes their eyes far more interesting. Once you get to something like a shark, all right, oh, I'm sorry, we're still in lampreys, my fault. So that was a lancelet. It does not have a complex eye, but here's I'll go. I just go through this again with the lamprey. Here's a video just on the lamprey eye. This is a pretty complex eye. It's still simpler than what you would see in the jawed vertebrates. So for example, here you see that corneal muscle. The corneal muscle pulls on the cornea to push the lens back or forward. And so thus it is that which, changes the focus rather than having ligaments attached to a ciliary body, which change the shape of the eye. By the time you get to the jawed vertebrates, the eye is pretty standard by this point. So the jawed vertebrates, they make a much larger eye. There's smooth muscle in the iris. There are primitive eyelids, etc. So if you were to look at a shark's eye here, there's the lens, the cornea, and the iris there. There's a ciliary body. Although it doesn't have muscles. So once again, they are moving the lens to change the position, the focus of the eye, rather than pulling on the lens to change its shape. So then, as I mentioned, those sarcopterygian fish, as they started adapting to life on land, they moved the position of the eye. So they were still fish, but seeing on land, was becoming, important enough that there was selective pressure to change the position of the eye. There are glands which moisten the eye in the first tetrapods. Obviously, being on land, that's important. When we look at mammals, most mammals are nocturnal. And so mammals have an awful lot of rods in their eyes in general. But the primates, as they evolve, would place more and more importance on eyes in general, and also then cones. And so, which would allow for color vision. So as we look at one of the defining features of the primates is the post-orbital bar, which surrounds the eye and bone to protect it. And then as we go in the anthropoid primates, they have bone behind the eye. So eyes are becoming more and more important. And so the bony protection of it becomes more important. Obviously mammals with their hair, this is where we'll get eyelashes, eyebrows, etc. The visual cortex of the cerebrum in primates starts to develop more so than in most mammals, and that's as we go through primates to anthropoid primates, etc. The parts of the brain involved in vision are getting bigger. Something very important happens in the ancestor of the catarrhine primates, that would be the old world monkeys, apes, and humans, that where there was once a gene for a cone opsin on the X chromosome, which perceived red and green light. There was a genetic mistake, a duplication. The gene duplicated so that now there were two where there was once one. And then over time, these changed slightly. So instead of perceiving both red and green, one of these got a bit better at perceiving red light, one got a bit better at perceiving green light. And so this made primates trichromatic, where they could distinguish between red and green, unlike most mammals, which are dichromatic. They can distinguish between red and blue, but they can't distinguish between red and green. And obviously, if you're living in trees, berries can be red, the leaves are green, that's important. And so a lot of the, primates, gradually increase the relative importance of their eyes. Now, eyes and vision, very complex. And I have a bunch of videos on that, but I'd like to just touch on one other visual topic. The ancestor of the eye was a photoreceptive brain. And the retina portion was not the only photoreceptive portion. In the diencephalon of the brain, there was a region called the pineal gland, forming part of a pineal organ, that was also receptive to light. Now, we think of the pineal gland as what makes melatonin at night. So you still have a pineal gland, and every night it's making the hormone melatonin, which is really important to you. So it's part of this daily light dark cycle, all right, this pineal gland in your body, but you don't really associate it with light. But the interesting thing is the cells of the pineal gland which are making melatonin are called pinealocytes. They have a common ancestry with rods. So if you look at primitive chordates and the photoreceptors that they have, where this primary cilium is now developing these compartments with opsins, it was from primitive receptors like this that both rods come from and these pinealocytes, which are photoreceptors in the brain. Now, I know that sounds odd because we mammals, our pinealocytes, that primary cilium is really small, and does it detect light? No, it doesn't. But when you look at the lamprey pinealocytes, the pinealocytes of fish, amphibians, reptiles, and birds, do they detect light using opsin proteins locating on the outer segment of a modified cilium? The answer is yes. So these pineal photoreceptors share a common ancestry and common structure with the photoreceptors in the eye. Now you're thinking, but wait, pinealocytes, they make the hormone melatonin. Yes, and that's also true of rods. And these pinealocytes in other organisms, your rods make melatonin. It just doesn't function as a circulating hormone which goes into the blood. It functions locally. So once again, the brain developed the ability to perceive light, and some parts of the brain then became more specialized for its perception. Not only the retina, which would then separate from the rest of the brain, but also then this structure called the pineal gland in the diencephalon. Now, one of the reasons is, so here in mammals, we have such a big cerebrum that it covers the diencephalon, but in most of the vertebrates, the pineal gland is actually exposed at the dorsal surface of the brain. So having a pineal gland, which is on the surface of the diencephalon and can perceive light, is a shared feature of the of the vertebrates. Now, in mammals, our great expanse of the cerebrum covers it. It ends the midbrain exposure. All right, so see, here's the human pineal gland. Notice it's covered. Here in sheep, it's covered. So it was no longer able to perceive light. So now what happens in your brain is light comes in through your eyes, goes to the hypothalamus, and the hypothalamus tells the pineal gland when to make its melatonin. But in more primitive vertebrates, the pineal gland at the top of the head, it was sensing when light was present. It sensed light or dark because light came into the top of the head, and the pineal gland would then control biological rhythms, sensing light directly. And so the photoreceptive brain gave rise to the retina of lateral eyes, but it also gave rise to a photoreceptive area known as the pineal gland. Now, one of the things you might be thinking is, wait, how can light come in the top of the head? Well, because there was a hole. All right, the primitive vertebrates, they all had a hole between what would become the parietal bones, allowing light to come into the pineal gland. Once again, not for vision. There's more things to do with light than just see with it. Once again, detecting day or night, detecting the length of the day, which lets you talk about seasons. These are various things. So the fish let light come into the top of the skulls. The fish ancestors of amphibians had a pineal foramen. The first amphibians had a pineal foramen. The first reptiles had a pineal foramen. Even the ancestors of mammals, this pelycosaur synapsid, my fault, this therapsid synapsid still had the pineal foramen. It would not be until the last step before mammals, the cynodonts, that the pineal gland was lost. The earliest bony fish known in the fossil record, last I checked, had a pineal foramen as well. And so the vertebrate skull was designed to let light come in the top of the head. So there would have been a space between skull bones. There would have been an area of translucent skin, and light came in to strike this pineal gland. And so it would have then functioned in light perception. And we can still see this today. So if you were to look here, there's a translucent area here on this lizard skull. If I go back to some of the lamprey images, you'll see that there's a transparent area there. Let me go back to. So if you were to look at the head of a lamprey, notice at the very top of the head, there's a little gray region where there's translucent skin. Once again, this is to let light. So right there, that lets light come right there. Notice light is coming in through a translucent area, and there's a photoreceptive part of the brain right underneath that. So light perception can involve more than just vision. There's lots of reasons to perceive light. Amphibians, they detect light with their skin, so that's more complex. So I'd like to wrap up, but just saying, vertebrates vary. There are actually more senses in the vertebrates. So a number of vertebrates, their eyes can detect ultraviolet light, like birds can. So just as mammals had more opsons, which allowed them to distinguish, or primates, between red and green, birds and other some fish can detect ultraviolet light. There are snakes which can detect heat. All right, so the pit vipers have pits here, which allow them to hunt warm-blooded prey, like the rodent that you saw in the previous image, because at night they can detect very small changes in the amount of heat. So that is a sense that vertebrates can possess the ability to sense heat. Some fish are receptive to electrical stimuli, and we could get into a conversation about electrical fish. I mentioned that bats are using high frequency sound waves to steer. Well, some birds do that, and the aquatic mammals also use this echo location. And so obviously, if you were to specialize, you could speak about a lot of these different vertebrate senses. But this lecture was just kind of meant to be an overview, primarily on the special senses in vertebrates and how they function in humans.